This video is going to explore three related topics: 7.4, 7.5, and 7.12. Population genetics, Hardy-Weinberg equilibrium, and variations in populations. An individual's phenotype is largely based on its genotype, the combination of alleles for each of its genes that it obtains from its biological parents collectively determine its overall phenotype. Since an organism's genotype doesn't change over the course of its life, natural selection's action is on the population as a whole, not on a given individual. Natural selection drives changes in the proportions of alleles found within a population. A given allele in a gene pool may, over generational time, become more common if it is selected for, or less common were it to be selected against. Additionally, non-selective, random events and changes to environmental conditions also affect allele frequencies in a population's gene pool. The amount of variation found within the gene pool is directly related to not only that which natural selection acts upon, but it is also important in a population's resiliency in the face of changing environmental conditions and its potential longevity. We know through Darwin's work, and also just by being observant in nature, that there is variation between individuals' phenotypes within a population. The importance of these variations cannot be understated. It is variation that provides the options from which natural selection chooses. Some variations are favored and are therefore more fit, whereas others are less so and are disadvantageous. When populations have minimal variation, individuals are more alike in phenotype. Depending upon the agent of natural selection, this means there may be a disproportionate number of individuals being selected for or against. For example, if a population had very little variation and was genetically highly similar, a greater number of individuals might be susceptible in the same way to a pathogen. By definition, a population is a group of individuals all belonging to the same species that are interbreeding with one another. The aggregate of all alleles possessed by individuals within that population is its gene pool. In a population of 30 individuals, a total of 60 alleles exist for a given gene, since each individual is diploid. Some individuals have 2 dominant alleles, others have 2 recessive alleles, and yet others have 1 dominant and 1 recessive allele. When studying evolution, the frequency or commonness of those alleles is represented symbolically. Lowercase and typically italicized p represents the frequency of the dominant allele. Lowercase italicized q is used for the recessive allele. Mathematically, the sum of a gene pool's frequencies of alleles is set equal to 1. The proportion of the dominant alleles, represented as a decimal, plus the proportion of all recessive alleles equals all alleles, 100%, or simply 1. Using binomial expansion, the allele frequencies equation is useful in estimating the genotypic frequencies of a given gene for a population. P squared plus 2pq plus q squared equals 1. A Punnett square helps to graphically illustrate the relationship between allele frequency and genotype frequency. The individuals with 2 dominant alleles, p squared, are homozygous dominant. The 2 individuals that are heterozygous have 1 of each allele. And q squared represents those possessing 2 recessive alleles. When analyzing or solving problems involving the Hardy-Weinberg equation, the proportion of individuals in a population that are homozygous recessive is typically the most readily useful information. You may recall that this is because, when observing a population, only the homozygous recessive individuals have a unique phenotype, the recessive one. visual observation of individuals with the dominant phenotype is insufficient for determining whether or not they are homozygous or heterozygous. The Hardy-Weinberg principle and equation is named after G.H. Hardy, a mathematician, and Wilhelm Weinberg, an obstetrician, who both independently mathematically illustrated it. Interestingly, Hardy's work focused on debunking the idea that a dominant allele would naturally tend to increase in frequency. At its essence, the principle states, allelic and genotypic frequencies will remain unchanged over generations if not acted upon by evolutionary forces. If a population's allele frequencies aren't changing, it is said to be in a state of Hardy-Weinberg equilibrium. Consequently, the genotypic and therefore phenotypic frequencies in a population will also remain unchanged. Let's analyze this graph to explain that concept. Let's say that a population has a dominant allele frequency of 0.33. The red line tells us that approximately 10% of the population would be homozygous dominant. Where the green and blue lines intersect, at 45%, tells us the frequencies of both the heterozygous and homozygous recessive phenotypes. If that population's allele frequencies remained at 0.33 for dominant and 0.67 for recessive, its genotypic and phenotypic frequencies wouldn't change either. The population is not evolving. But in order for allele frequencies to remain the same over generational time, 5 conditions must be satisfied to maintain the state of Hardy-Weinberg equilibrium. And here they are, presented in no particular order. First, the population must be large. The smaller the population, the more likely that its allele frequencies will fluctuate due to chance events from one generation to the next. This is a process called genetic drift, which we'll take a look at in a few moments. Next, mating must be random. This means that individuals cannot demonstrate any preference for potential mates. If an individual does express preference for potential mates, then that is a form of selection. Third, there can be no migration. Also known as gene flow, which will come up in a few moments as well, when individuals immigrate into a population or emigrate out of it, they are bringing in or taking away, respectively, the alleles they possess. Next, there can be no natural selection. This means that all individuals in a population have equal fitness and an equal probability of survival and reproduction. Natural selection results in differences in the survival and reproductive success of individuals carrying advantageous variations. And finally, there can be no mutations. A mutation is a random permanent change to an organism's DNA. Mutations are also the singular source of novel alleles in a gene pool, and is therefore modified if mutated alleles appear. If any one of those 5 requirements is violated, then allele frequencies can change. A change in the gene pool of a population results in shifts in genotypic frequencies and ultimately the phenotypic makeup and variations and their proportions present in a population. You may be wondering, since the conditions for a population to remain in Hardy-Weinberg equilibrium are almost impossibly unlikely to occur in nature, what's the point? How can such a principle and its associated equations be useful? One application of the principle is its use in determining the speed or pace of evolution in a population. If the allele frequencies of a population's gene pool are monitored over many generations, how much they change is an indication of things like the selective pressures and the organism's environment. The proportion of individuals in a population that are carriers for a given trait or disorder can be estimated. Genotypically speaking, after all, carriers are heterozygous, 2pq, And in a controlled laboratory setting where 4 of the 5 conditions can be artificially maintained, the rate at which mutations occur can be studied. With a large population of study animals that are confined, in which all individuals have an equal probability of mating and survival, the only source of change ought to be the introduction of new alleles, mutation. Natural selection acts upon an individual's phenotype. Depending on which phenotypic variations are selected for or against, that will alter the makeup of the overall population in certain ways. Observe in this model the phenotypic variation present in a population of mice. The extreme variations, the very lightest and very darkest fur colors, are the least common, whereas the intermediate phenotypes are most common. If natural selection forces were to select against one of the extreme phenotypes in favor of the other extreme phenotype, that is referred to as directional selection. This example happens to illustrate the selection for darker fur. Over generational time, the phenotypic makeup of the population would be comprised more and more heavily of mice with darker fur. On the other hand, if the intermediate variation was selected against, that would result in diversifying or disruptive selection. When populations are subjected to persistent diversifying natural selection forces, this can result in groups of individuals evolving such great differences that they are no longer capable of interbreeding with their alternate variation counterparts. This phenomenon of speciation will be studied in a later video. Finally, when both extreme phenotypes are selected against, and the intermediate variation is selected for, this is called stabilizing selection. Stabilizing selection reduces the variation and the genetic diversity within a species. So far, the mechanisms behind evolution, natural selection, sexual selection, and artificial selection have all been non-random in nature. Individuals are selected for or against for particular reasons. Some have advantageous variations, others have deleterious variations. Those that are selected for are said to have a greater fitness and are therefore more likely to leave offspring, and perhaps a greater number of them. However, there is a wide-ranging collection of events, occurring randomly, that influence the allele frequencies of a population. When events are random, an individual's fitness plays no role in its survival or reproductive ability. When a population is small, chance plays a bigger role in influencing the allele frequency of a subsequent generation. Chance events cause allele frequencies to fluctuate unpredictably from one generation to the next. Occurring by several mechanisms, genetic drift is non-selective. Consider this small wildflower population. It has a stable size of 10 plants. Suppose that, by chance, only 5 of the initial generation produce fertile offspring. Perhaps this occurs because only those plants happen to grow in a location that provided enough nutrients to support the creation of offspring. Again, by chance, only a couple plants in the next generation leave fertile offspring. The result, after only a few generations, is a dramatic shift in allele frequencies, in this case completely eliminating the recessive one. The second cause of genetic drift is called the bottleneck effect. A sudden change in an environment, such as a fire, volcanic eruption, or flood, that drastically reduces the size of a population. This severe drop is referred to as the bottleneck effect because the population passes through a bottleneck that reduces its size. By chance alone, certain alleles may be overrepresented among the survivors and others underrepresented, and some may be entirely absent. In this model, if each marble color represents an allele in a gene pool, a random destructive event has managed, by chance, to completely eradicate the yellow allele in the population. Additionally, we can qualitatively estimate that the blue and white alleles were originally in roughly equal proportions, but in the surviving population, 5/6 of the alleles are blue. When a few individuals become isolated from a larger population, this small group can establish a new population in a previously uninhabited area by that species. This is called the founder effect. It might occur, for example, when a few members of a population are blown by a storm to a nearby island. Those individuals and the alleles they carry with them are used to establish, or found, a new population. Depending upon the genetic makeup of the founders, its population may look very different from its original ancestral population. With any of the mechanisms behind genetic drift, there are 4 key points to remember. The first is that genetic drift is significant in small populations. Second, allele frequencies can change at random, not being influenced by selection. Third, it often leads to a loss in genetic variation. And fourth, the potential exists for harmful alleles to become predominant, threatening the population's survival. Genetic drift is not the only non-selective phenomenon affecting allele frequencies. They can also change by gene flow, the transfer of alleles into or out of a population due to the migration of individuals. When gene flow is high, and the transfer of genetic material between populations occurs regularly, the allele frequencies in different populations tends to be more alike. This is an important mechanism for transferring genetic diversity among populations, and is thought to constrain speciation. Gene flow is also important in introducing a previously existing allele to a population that doesn't currently have it in its gene pool. This is exemplified by a homozygous recessive individual migrating into a population of entirely homozygous dominant individuals. When gene flow is prevented, perhaps due to a natural event, like a river slowly carving out a canyon, or an artificial one, like highways being built that fragments a habitat, the genetic differences between the isolated populations tends to increase. Over time, because of alternative selection forces, favoring some variations and selecting against others in each of the isolated populations, differences may become so great that they are no longer capable of interbreeding. They're no longer the same species. Mutations hold a special place in evolution. They are the only mechanism by which novel alleles appear in a population. From the perspective of the Hardy-Weinberg principle, it means a third allele is included in the allele frequencies equation. And, just as before, through binomial expansion, the variables for genotypic frequencies are established. A great, well-known real-world example is human blood types. In this case, the P and Q represent equally codominant alleles, A and B, and the R represents the recessive O allele. Each of the genotypes has a corresponding variable for its frequency in a population. It's also important to remember that new alleles arising by mutation may or may not generate new phenotypic variation and may or may not increase fitness. Consider the rock pocket mouse. The mutation for dark fur, a disadvantage on a light background, but a clear benefit on a dark background. The mutation for blue eyes in humans, a disadvantage in environments with intense solar radiation, but neutral and perhaps an advantage in other environments. When mutations occur in bacteria, resulting in greater resistance to antibiotics, that's clearly an advantage for them. And that brings this exploration of these 3 topics to a close. As usual, thanks so much for watching, and take care.